BSc. Friends, today we are going to discuss the fundamental principles of inheritance which were explained by Gregor Johann Mendel. Today we are going to discuss the monohybrid cross and its principle, and the dihybrid cross and the principle deduced from dihybrid cross by Mendel. Let's get started. Mendel laws of inheritance. Today's contents are introduction, terminology, monohybrid cross, and dominance and its definition, law of segregation, dihybrid cross, law of independent assortment, test cross, references. Friends, you know very well. Doing research in genetics is very difficult because everything is an imagination. Lot of mathematical or statistical data is to be collected. Mendel did all these things for us. But before Mendel also, several scientists like Kohlreuter and others worked on genetics, but they could not succeed because the approach of genetical research is totally different from that of anatomical research or physiological research. We need to involve lot of statistics to find out the correct pathways or correct principles. So Gregor Johann Mendel succeeded in this pattern. Gregor Johann Mendel is now considered as the father of classical genetics. He was born in Austria, Moravia in Austria, which is presently in Czechoslovakia. So he was born on July 22nd, 1822. He was actually, after having completing the graduation, so he joined in Augustinian monastery and he served there as Austrian monk. But while working in Augustinian monastery, so he developed a monastery garden with the pea plants and conducted a lot of experiments of inheritance on these pea plants during 1856 and 1863 and discovered the basic principles of inheritance or heredity. But the principles what were explained by Gregor Johann Mendel were in the form of statistics. They were in the form of ratios. Contemporary scientists could not understand what are these things. Okay, so simply they were not they did not care about these principles. Okay, so almost all the discoveries of Mendel were degraded. They remained under cover. But when Charles Darwin proposed his Darwinism, so the scientists need what is the actual process of inheritance, because Charles Darwin explained the process of inheritance in a wrong manner. So to make the Darwinism survive, Mendel principles were added to Darwinism by the disciples of Charles Darwin. So Mendelism was rediscovered by Hugo de Vries, Karl Correns, and Erich Schemack. Of course, a scientist by name Bateson popularized Mendelian genetics. Mendelian genetics was popularized by Bateson. Bateson himself coined the word genetics for this kind of study. This was a new branch at that time, and the name of the branch is coined as genetics by T.H. Morgan. And some other scientists like Fisher, Haldane, and Sewall Wright. So they considered Mendelian genetics to explain the process of evolution also. What are the statistical principles that were used by Mendel also applied to explain evolution pattern, especially the modern synthetic theory of evolution, totally dependent on the Mendelian principles. So, how was Mendelism rediscovered? So, a very interesting story. So, Mendel, in his Mendelism, explained that for every character there are two factors, and these two factors get segregated into the gametes. This was his main theory. So When Sutton and Boveri were working on the behavior of chromosomes during meiosis, they also explained that the two homologous chromosomes get segregated into gametes. Later, T.H. Morgan also proposed chromosomal theory of inheritance. So all these attracted the scientists towards Mendelian principles. So in such a way, Mendelian principles were appreciated later by almost all scientists. Okay? So Mendelism has paved the way for Darwinism, population genetics, modern synthetic theory of evolution. So friends, it's very important to know about the basic principles explained by Gregor Johann Mendel. Mendel's experimental material was the pea plant, that is Pisum sativum. He selected many characters in that, but he got what is that called prudent results only from these 7 characters. 7 characters in pea plant were selected by Mendel for his experiments. They are color of the seeds or color of the cotyledon. There are 2 forms. One is yellow color and green color. Flower color is white or purple. That may be seed coat color also. So ripe pod is shape of the ripe pod is inflated or constricted. Color of unripe pod is yellow or green. Height of the stem is tall or dwarf, and form of the ripe seeds, round seeds or wrinkled seeds, and the position of flower, it may be either axial or terminal. So what I am describing here, so first yellow, later green, indicating that the first one is a dominant trait and the second one is the recessive one, following the recessive. Dominant white, recessive. Dominant inflated, recessive constricted. Yellow is dominant here when compared to the color of unripe pod, whereas the seed color yellow is dominant here, but here green is dominant. Green is dominant and yellow is recessive. Here tall is dominant and dwarf is recessive. Round is dominant and wrinkled is recessive. Like that, axial is dominant. So, okay, these many characters in a pea plant were studied by Mendel to what is that called, explain the basic principles of genetics. Only Mendel could succeed, but other scientists could not succeed. Why? What is the reason? There are many reasons. Mendel did the scientific research in genetics in what way actually needed. In the similar way, he conducted the genetic research. Other scientists concentrated many characters at a time. But Mendel did not do like that. Mendel could concentrate only on one character or few characters at once. So he wanted to understand the inheritance process of one character only. What is the inheritance process of one character? If one character's inheritance process is like this, and what are the inheritance process of 2 characters? And is there any relationship between these 2? So what are the inheritance procedures of 3 characters at a time? Is there any relationship? Like that, meticulously conducted experiments, and so that is why the idea of Mendel made Mendel success in his experiments. What was the idea? So every character must be studied separately to understand the basics. Okay, so he conducted experiments meticulously, and this is very, very important that every data so recorded properly to understand the relationship between the numbers. Mendel was the first person to say about factors. Okay, unitary factors, we can say. Every character will be flowed to the next generation or inherited to the next generation by means of some factors located somewhere in our body. Later, they were discovered that they are located in chromosomes and they are nothing but the genes. So his idea that there are some factors. And then he was the first person to discriminate the traits from characters. Say, let me tell you, height. Height is a character. It is having 2 traits. One is called tall, the other one is called dwarf. So this was his idea. Actually, other scientists thought that a tall was one character and a dwarf was a separate character. But Mendel could understand it properly that every character is appearing in two alternative forms. Likewise, every gene has two alternative forms. Okay, these alternative forms of a gene means it is later coined by Johansen, of course. So the two alternative forms of a gene what were explained by Mendel are called alleles. Mendel explained that every character is flown by two alternative factors. They are called alleles. Okay, so this idea of alleles and this idea of traits was there only with Mendel. That's why he could succeed in the genetic experiments. Of course, selecting choice of Pisum sativum, selecting Pisum sativum is also a very, very important factor bringing success to Mendel, because it is the plant which is having a lot of advantages. It is an annual plant. So within a short span of time, we can get the results. And it is a bisexual. Okay. So that self-pollination is possible so that the pure breeds are available. And it is a plant where cross-pollination is also possible. Emasculation can be done very easily. So, and it is the plant, it is the species that is having all these 7 characters on 7 separate chromosomes. So, no question of linkage in these characters what are selected by Mendel. So, of course, that may be a lucky factor. That might be a lucky factor behind the success of Mendel. Okay. So, one of the reasons why Mendel could succeed is he analyzed the data in a statistical manner. So, all other scientists wanted to draw the diagrams, but here a different approach he has chosen and laid the foundation stone for developing genetics as an emerging branch. Okay, so there was no linkage. So that's why that is also one of the reasons of Mendel's success. Anyhow, Mendel succeeded. Let us see how he could conduct the experiments. To understand Mendelism, one has to know about certain terms what we are using in this lesson. Okay, hereditary units. These are the small factors, nothing but gene. They are nothing but genes. Actually, later they were termed as genes. These factors are causing inheritance according to Mendel. Next, alleles. Alleles are nothing but two alternative forms of factors, of which one is expressing immediately, that is called dominant allele, and one is not expressed in the presence of the other, that is called a recessive allele. Allelomorph. Allelomorph means the alternative forms of a character. Height is a character which has two allelomorphs, tall and dwarf. Shape of the seed, it's a character, it has two allelomorphs, round and wrinkled. So likewise, every character has two traits. Okay, and every character is transmitted by two factors, a dominant allele, a recessive allele. Okay, phenotype. These were the first. These were the terms first used by Mendel, and still we are using the same terms. So such has been the purity of the discovery what he made. Okay, phenotype. Phenotype is simply external appearance of a character. Say a plant is tall, tall is phenotype. A plant is dwarf, dwarf is a phenotype. And what is genotype? Genotype is the genetic constitution of a character. You take for example a plant is tall. What is responsible for its tallness? The responsibility is taken by capital T genes. Alleles. Capital T is the genotype. Capital T small t will also express in the form of tallness. So that is its genotype. A tall plant is having capital T small t, two genotypes. A dwarf plant is having only one genotype, that is small t. Okay, genotype, genetic constitution irrespective of its phenotype. Phenotype, external appearance irrespective of its genotype. Homozygous condition. When the two alleles are of same kind, such a condition is called homozygous. Capital T, homozygous. Heterozygous. When the two alleles of the same character are different, such a condition is called heterozygous condition. For example, capital T, small t. Then monohybrid cross. Monohybrid cross is an experiment, a hybrid cross which is conducted between the two traits of a character where the inheritance of only one character is studied. The inheritance of only one character is studied in an experiment. That experiment is called monohybrid cross. Dihybrid cross. What is dihybrid cross? The inheritance of two characters simultaneously studied. You take for example yellow color of the cotyledon and round shape of the seed, two characters, and it's contrasting, green and wrinkled. Two characters are studied at a time and to find out the inheritance pathway. That is called dihybrid cross. Then test cross. What is test cross? Test cross is the cross that is conducted to identify the genotype of a dominant phenotype. Earlier, just now I mentioned that a dominant phenotype is having two genotypes, capital T or capital T small t. To find out what is its actual genotype, we need to conduct a cross. That cross is called test cross. It is conducted between the F1 hybrid with its recessive parent. And ratios. Mendel explained everything in the form of ratios. Monohybrid phenotypic ratio 3:1, monohybrid genotypic ratio 1:2:1. Like that, he went on explaining every result in the form of ratios. Let's go on to the next slide. Friends, this is a chart I developed to make you understand about monohybrid cross. What is a monohybrid cross? It is the cross which is conducted between the two traits of a character to identify their inheritance phenomenon. Only one character is studied, and one character has two alternative forms. So I have taken here in this slide character height. I have taken character height. This character height has two alternate forms. Two allelomorphs are there. One allelomorph is tall and another allelomorph is dwarf. Okay. So tall is called its phenotype. Tall is called its phenotype. Phenotype means external appearance. It is tall, that's why it is phenotype. It is dwarf, that's why it is phenotype. What genes are there inside we don't want. So only external appearance is explained by phenotype. And what are the genotypes? Because for the P1 generation, parental generation, Mendel took only the pure breeds. Mendel took only pure breeds. What are pure breeds? Pure breeds are homozygous. Homozygous means capital T and also a capital T. Dwarf plant is always homozygous. Small t. And how to develop a pure breed? Very easy. According to Mendel, if any plant is taken and it is self-pollinated for 5 generations. If it is self-pollinated for 5 generations and we select only tall plants for the 5 generations and we cull out all the other plants, then we'll get the pure breed. So in such a way, everything, every point, every moment, Mendel took a lot of care and conducted the experiment. So that's why he could get the result. Okay friends, let us see a tall plant, a dwarf plant. Tall plant genotype is homozygous. Dwarf is also homozygous. Tall is capital T, dwarf is small t, I'm sorry. And this tall plant, according to Mendel, the two factors capital T get segregated, got separated. And here small t gets segregated, got separated according to Mendel. These are all the ideas of Mendel. Now, the segregated factors of one plant crossed with the other one. So, cross-fertilization occurs. So, when capital T and small t fertilized, then what happens? Come together in a generation, in an individual. This individual is called capital T, small t. Capital T, small t. capital T, small t, capital T, small t. So, when Mendel took a tall plant and crossed it with a dwarf plant, in the F1 generation, first filial generation, he could get all tall plants. He wondered. So, he took two different plants, but he got only a single phenotype, that is tall. What might have happened to the dwarf? He conducted this experiment for many times, and all the time he could get only one result, that when tall and dwarf were crossed, pure plants were crossed, in the next generation only one phenotype will get. So that phenotype is having heterozygous genotype. This is heterozygous. Now he wanted to test the purity of these tall plants. In F1 generation he got the tall plants, okay, and in P1 generation also there is a tall plant and both are having same height. So he wanted to test whether these tall plants are real tall plants. So to find out that, so he conducted what is that called self-pollination. Self-pollination, that means capital T and capital T of same genotypes. So making a cross between two same genotypes is called self-cross or selfing, we say. So the two factors you see, capital T, small t. Here also capital T, small t. Then what has happened? When capital T combined with the capital T, we will get capital T, capital T, small t, and small capital T, small t, small t, small t. So this generation is called F2 generation. When he observed the phenotypes of F2 generation, tall, he could get 3 tall plants out of 4 and 1 dwarf plant out of 4. So what is the phenotypic ratio? 3 tall, 1 dwarf, 3:1. So 3:1 is the phenotypic ratio. And how many genotypes are there? Let's see. Capital T, homozygous dominant genotype. Capital T, small t, this is heterozygous genotype, homozygous recessive. Homozygous dominant is 1, heterozygous are 2, and homozygous recessive is 1. So what is the genotypic ratio? 1:2:1. So for many times he repeated the experiment with other characters and other plants, he could get the same ratios. So that's why, based on the results of this monohybrid cross, he proposed certain principles, of which the first principle is called law of dominance. Of course, this law of dominance is not suitable for some characters and is suitable only for, is applicable or suitable or correct only for some characters. When two different traits are crossed, out of the two traits, only one trait will be expressed in the next generation. The expressed trait is called a dominant trait. And the factor responsible for this trait is called a dominant factor. Okay, when two traits are crossed, only one will be expressed. The expressed character is called a dominant character. Okay, what is not expressed? We are crossing the pure breeds, keep it in mind. We are crossing the pure breeds, then only what I am saying is correct. So only one will get, that is called dominant. The other character is not expressed here. It doesn't mean that there is no such character, but it is there and is suppressed. The suppressed character will be expressed in the next generation. That means whatever character is suppressed is called a recessive character. Mendel proposed this theory or this law of dominance. But it is not considered as the first law of dominance because it is not correct or not true for all the cases. There may be codominance, there may be incomplete dominance in other cases. That's why, but this is the truth. There is the dominance. One out of the two factors, one factor is dominant, one factor is recessive. Out of the two forms of a trait, a character, one trait is a dominant trait and the other trait is the recessive. Earlier in the cross, what we have seen, tall is the dominant and a dwarf is the recessive character. Okay. Now, Dominant character will be expressed in homozygous condition and also in heterozygous condition. That is the problem. Whether a dominant individual is there, we don't know whether it is having homozygous genotype or heterozygous genotype. That's why test cross is needed. But whereas the recessive character is there, recessive character is always homozygous. Right. So this is first explanation given by Mendel towards dominance. Let's see the first law of inheritance. The first law of inheritance is called the law of segregation. Law of segregation. Law of segregation is also known as the law of purity of gametes, and it is also known as non-alignment theory of gametes. What is law of segregation? The two factors which are responsible for one character tend to segregate or separate, enter into the gametes. And these segregated gametes may be reunited in the next generation. So when they got segregated in one generation and maybe reunited in the third generation, and during this course of segregation and reunion, one factor will not exchange its character to the other factor. The factors always remain pure in the gametes. Very simple. There are two factors for a character. You see here I am explaining capital T, capital T. These two factors get segregated. They get segregated, separated into separate gametes. Okay. You see capital T, small t. Capital T entered into one gamete and small t entered into the other. This is called segregation. Okay. Small t, one small t entered into one gamete, another small t entered into another gamete. This is called segregation. But what is very, very important here is when the gametes segregate, when the factors segregate into the gametes and stay with the other factors there in the particular individual, as long as they are staying with the other factors, they don't exchange their characters. They remain always pure. Because they are remaining pure in the F1 generation, after reunion in the F2 generation, the same trait of P1 generation is expressed. Let me explain that here. You see here, to explain this purity of gametes, I have drawn a red color for capital T, and a black color for small t. Let us see these two factors segregated. Okay, segregated. The segregated capital T is living with small t, segregated small t in one generation. Capital T is there with the small t, but capital T did not exchange its character with the small t, or small t doesn't give its character to the capital T. There is no exchange of characters between these two. Now what is happening? You see, these two got segregated. Okay, in this generation, the capital T are reunited here in the third generation. From first generation, they got segregated into the second generation. P1 generation segregated into the F1 generation, but reunited in the third generation. So let us see the height of this tall plant is the same or different to that of P1 plant. Mendel, measured the length of these two plants. So first he recorded the length of that plant and he recorded the height of this plant. Strangely, he found that both the plants are having same height. That means capital T, when is there with a small t in one generation, during that time it remained pure without exchanging its character with small t. Likewise, the dwarf plant of P1 generation and dwarf plant of F2 generation both are having same height. It is possible only when the factors always remain pure in the middle generations. That is why it is called the law of purity of gametes. Okay friends, let's go on to the next slide. Mendel's dihybrid cross. Mendel conducted dihybrid cross to find out the inheritance pattern of the two characters simultaneously. One character he already found as that it is resulting phenotypic ratio 3:1, sorry, and genotypic ratio 1:2:1. When he conducted a dihybrid cross, that means find cross conducted to find out the inheritance of two characters simultaneously. So strangely, he could find that a dihybrid cross is a product of two monohybrid crosses. Exactly, it is following the statistical principles. You take the monohybrid, what is that called, phenotypic ratio 3:1. So you multiply 3:1 into 3:1. You will get 9:3:3:1. So it is a very, very interesting factor that Mendel could explain about the dihybrid cross, which is a product of two monohybrid crosses, and likewise trihybrid cross. So he went on explaining like that. So there is a definite mathematical or statistical approach in genetics. That's why population genetics developed by these principles. Okay, Mendel explained the recombinations. What is the reason for the recombination? Actually, in meiosis, the chromosomes will be broken down into pieces, and the piece of mother chromosome attached to father chromosome, vice versa. Because of that, recombinations occur, and because of those recombinations, Mendel Mendel explained here law of independent assortment, which is proved correct by the recombinations. Okay, let's go into the dihybrid cross mechanism. Let's see Mendel dihybrid cross. Okay, so P1 generation phenotypes, he selected yellow character, round character simultaneously, and its counterpart, contrasting form. Yellow contrasting form is green, and round contrasting form is wrinkled. So he selected two plants. One is having two characters, yellow and round. The other is also having its counter characters called green and wrinkled. So he thought that yellow is because of capital Y, round is because of capital R pure breed. And here green is because of small y, and wrinkled is because of what is that called, small r. And he deduced the gametes, that is capital Y, capital R, here small y, small r. When these got fused, all are yellow and round, capital Y, capital R, small r. Okay, he wanted to make a self-pollination by conducting emasculation. So he considered one as a male and the other one as a female. Friends, you just observe here how many different types of gametes are produced by a dihybrid. A dihybrid, this is dihybrid. Okay, this is not hybrid, this is a pure plant, but this is hybrid. A dihybrid is producing 1, 2, 3, 4 types of gametes. You see, these are all nothing but permutations and combinations. Capital Y enters into the gamete along with the capital R. Okay, forming a composition called capital Y, capital R. If capital Y enters with the small r, we'll get capital Y, small r. But what is the small y nature? Small y can enter with the capital R, can also enter with the small r if it is not possible. So all kinds of permutations and combinations can be resulting here to form 1, 2, 3, 4 types of gametes. So how many types of gametes are formed by a dihybrid? 4. How many types of gametes are formed by a monohybrid, F1 monohybrid? 2. Capital T, small t. Okay. So similarly, the female case. So let us arrange all male gametes here and all the female gametes here, and let us make a Punnett board. It is a very easy process. Checkered board or Punnett board. When capital Y, capital R, capital Y, capital R are fertilized, then we get capital Y, capital Y, capital R, capital R. And likewise, capital Y, capital R, small r, capital Y, small y, capital R, capital R. Likewise, capital Y, small y, capital R, small r, like this. All these are same genotypes. So this is a different genotype, this is a different genotype, this is a different genotype, this is a different genotypes. You see this diagonal. This diagonal is having all homozygotes, double homozygotes. Capital Y, capital Y, capital R, capital R. This is homozygote, this is homozygote. Double homozygotes. How many double homozygotes are there? 1, 2, 3, 4 double homozygotes are there out of 16. How many double heterozygotes are there? 1. So capital Y, small y is a heterozygote. Capital R, small r is also heterozygote. So 1, 2, 3, 4. 4 double heterozygotes, 4 double homozygotes are there. And the remaining all are homozygous to one character and heterozygous to other character. You see here, capital Y is homozygous, but capital R, small r is heterozygous. So, homozygous to one character, heterozygous to other character, 8 individuals are there out of 16. Friends, what is the percentage of double homozygotes? 4 out of 16, 25%. What is the percentage of double heterozygotes? 4 out of 16, that is 25%. What is the percentage of the genotypes which are homozygous to one character? Where is this? Homozygous to one character and heterozygous to other character. Okay, where is that? This is homozygous to one character and heterozygous to 8 out of 16, 50% are there. 50%. Okay, now in this checkered board you just observe out of 16 how many are yellow and round? 1, 2, 3, 4, 5, 6, 7, 8, 9. So this very big triangle you may be observing, I have given a different color to express the yellow and round. So all these are yellow and round, they are 9. And how many are yellow and wrinkled? You see the black color, 1, 2, 3 only, 3. How many are green and round? You see the green color, okay, light green color, green and round are 3. And you see another olive green color, yellow and wrinkled. I'm sorry, here small y, it must be. It must be small y. Just a minute, I'll change it now. Just a minute. Okay. Okay, now it is correct. Now, so so this is small y, small r. So this is green and wrinkled. Okay, so this checkered board will explain the phenotypic ratio as 9:3:3:1. Let me tell you, 9:3:3:1 is the phenotypic ratio. But what is genotypic ratio? Genotypic ratio means we need to identify the genotype, capital Y, capital R, or how many? How many individuals are having capital Y, capital R? If you can search, so I pointed out A, there is only one. B, you see B, here capital Y, capital R, small bar, here also the same genotype. So B are 2. A is only 1, B genotypes are 2. C genotype, here you see, and here the same. Right. Okay, just a minute. Small correction I'll make here. Small correction. Capital R. Okay, so that is yes. Now, okay. So these two are C, they are 2 genotypes. B, they are 2 genotypes. A only 1. 1:2, 2:2, 2:2. Oh, you see these are all 4. How many A? Only 1. How many B? 2. So 1:2, 2:2. C also 2, 2:2. 1:2, 2:2, 2:2, 4. And then how many are capital Y? You see capital Y. Small r, capital Y, small r. Just a minute, some. Yes, capital Y, small r. How many are there? So there are only one. There is only one. And how many are small y, capital R, only one. Okay. And how many are small y, capital R, 2? This is 1. Okay. Let me tell you, 1 is 2, 2 is 2, 2 is 2, okay, ABC. D, 4 is 2, 1 is 2, E, F, 2 is 2, 1 is 2, 2 is 2, 1. Friends, you see it carefully. 1 is 2, B, you see the B, 2 is 2, C, 2, D, 4, E, 1, F, 2, G, 1, H, 2, I, 1. So 1 is to 2 is to 2 is to 4 is to 1 is to 2 is to 1 is to 2 is to 1. So this is the genotypic ratio and this is the phenotypic ratio. And based on these results of dihybrid cross, Mendel could propose his theory called law of independent assortment. Law of independent assortment explains that one factor of one character enter into the gamete and can be assorted with any other factor of any other character. Factor while entering into the gamete feels as if there is no other factor at all. One factor can be assorted with any other factor. Assortment is dependent totally by chance. Let me explain here. You see here there are capital Y, small y, capital R, small r factors are there. Capital Y can be assorted with capital R or it can also be assorted with small r. You see here small y. Small y can be assorted with capital R or it can also be assorted with capital R. Just a minute. Yes, that's a small y. Yes, this is small y. Okay. Now, so this is called independent assortment. What is independent assortment? The assortment of factors in a gamete is totally independent. The assortment of different factors in a gamete is purely dependent on chance. Because of this independent assortment, recombinations are possible. Recombinations are possible. So, this is called law of independent assortment. Friends, what is test cross? Test cross is a cross that is conducted to identify the genotype of a dominant phenotype. Okay? When a dominant phenotype is crossed with its recessive individual, when we get ratio 1 is to 0, then the dominant individual is a homozygous plant. When a dominant individual is crossed with its recessive individual, when we get the ratio 1 is to 1, then it is heterozygous. So Mendel deduced a very simple cross to identify the genotype of an unknown, what is that called, a dominant phenotype. This cross is called test cross. And how is test cross performed? The F1 hybrid will be crossed with its recessive parent. That means it's a recessive charactered individual. It will be like this. You see, this is a tall plant. We don't know what is its genotype. This is dwarf plant. We know the genotype. It is always homozygous. So if the tall plant is heterozygous, when it is crossed with a dwarf plant, it produces 2 tall, 2 dwarf. So what is the ratio? 1 is to 1. 1 is to 1. Genotypic ratio is also 1 is to 2, heterozygous to homozygous. Okay. So when we get 1 is to 1 ratio, definitely the tall plant is a heterozygous individual. And if we get all tall plants like this, then the tall plant is a homozygous. The tall plant is homozygous when it yields all tall plants. A tall plant is said to be heterozygous when it gives 1 is to 1 ratio to tall and dwarf. So this is a very, very easy method to find out the genotype of a dominant phenotype. Okay, so these are the references I have referred to develop this video. Thank you, friends. Thank you for watching this video. Thank you.